There's one DNA fact that makes the simple version of ancient human history incomplete. The version most people carry in their heads. Europe over here, Asia over there, movement mostly west to east, cultures forming in neat geographic boxes. It feels logical, it feels intuitive, and it feels finished. But when you look at ancient DNA instead of stories, that clarity starts to dissolve. Because scientists found something unexpected in human remains from Siberia dated to around 7,000 years ago. Not a vague resemblance, not a weak signal, but clear genetic components that are usually associated with early European populations. And once that fact is on the table, the question is no longer whether our model is incomplete. It's how incomplete it really is. This video is not about shocking claims or rewriting history for effect. It's about assembling a picture that was always fragmented. We're going to build a genetic mosaic: paternal lines, maternal lines, ancient genomes, neighboring populations, and long-distance movement. When those pieces are placed together, they form a picture that textbooks rarely show, not because it's hidden, but because it's complex. And here's the strange part. The most unsettling detail is not simply that European-associated DNA appears in ancient Siberia. The real tension comes from how different genetic lines tell slightly different stories. One line points west, another doesn't. And that split forces a deeper question. When we ask, who were these people? Which answer are we actually listening to? That question is the loop that drives everything that follows. There are no heroes in this story in the usual sense. The closest thing we have to detectives are geneticists. And even they don't lead the story. The methods do. 3 tools matter here. First, the Y chromosome, passed from father to son, allowing researchers to trace paternal ancestry across thousands of years. Second, mitochondrial DNA, passed from mothers to all their children, offering a parallel maternal history. And third, ancient DNA itself, genetic material extracted directly from the bones and teeth of people who lived millennia ago. These tools don't speculate. They compare. They cluster. They reveal patterns that only emerge at scale. The Y chromosome tells us something very specific. Lines of descent that often move with groups of men. Mitochondrial DNA tells a different, equally valid story. One shaped by women, family structure, and long-term settlement. Ancient DNA allows these lines to be anchored in time, not guessed from modern populations alone. Together, they let us reconstruct population history with a level of precision that didn't exist even a few decades ago. Before we go any further, the rules of this investigation need to be clear. No myths, no cultural assumptions, no stretching data to fit modern identities. Everything we discuss comes from population comparisons, peer-reviewed analysis, and what the data can actually support. Where certainty ends, we stop. Where multiple interpretations exist, we acknowledge them. That discipline matters, because the finding we're dealing with is easy to misunderstand. When scientists say European DNA in a 7,000-year-old Siberian individual, they are not saying Europeans lived in Siberia. They are identifying genetic components that are most closely related to ancient populations known from Europe and Western Eurasia. These components form recognizable patterns, signatures shaped by shared ancestry far earlier than modern borders. And those signatures are real. They appear consistently across multiple samples. They align with known ancient genomes. They are not contamination. They are not statistical noise. They represent inherited ancestry from populations that were once connected across vast distances. The immediate reaction is to imagine a single migration, a group moving east, settling, disappearing. But the data resists that simplicity. What it suggests instead is interaction, repeated contact, gene flow over generations rather than the single dramatic event. Imagine small groups moving along river systems, following herds, trading resources, forming temporary alliances. Over centuries, these interactions leave traces, not in written records, not in monuments, but in DNA. Each generation carries a diluted but persistent memory of contact. That is what we're seeing in these ancient Siberian genomes. Archaeology hinted at this long before genetics could confirm it. Similar tool styles, shared technologies, overlapping subsistence strategies. But objects can travel without people. DNA cannot. Genetic signals require reproduction. They require families. They require time. And that's what gives this discovery its weight. What makes it more complicated is that this genetic signal doesn't dominate the genome. It blends. It mixes with local ancestry. It creates individuals who cannot be neatly placed into modern categories. These people were not European or Asian in the way we think today. They were something else entirely, the product of a world where human movement was slow, constant, and multidirectional. This is where many simplified narratives fail. They assume ancestry works like a straight line, but ancient DNA shows it behaves more like a web. Threads intersect, diverge, reconnect. Some persist strongly. Others fade but never disappear completely. And here's the critical point we'll return to later. If you only follow one genetic line, the story looks clean, almost too clean. But when multiple lines are examined together, that clarity breaks down. That breakdown isn't a problem. It's the evidence. In this series of blocks, we'll unpack what each genetic line actually shows. We'll see why the paternal story feels stable at first, and why that stability doesn't last. We'll explore how maternal ancestry introduces geographic patterns that seem to contradict expectations, but actually record something deeper. How populations form through mixing, not replacement. By the end, the question won't be, why is European DNA in ancient Siberia? It will be, why did we ever expect it not to be there? We'll start with the paternal line, because it offers the simplest answer. But that simplicity won't survive for long. We're not chasing legends. We're following traces in DNA. And the most direct place to start is the paternal line, the Y chromosome, because it behaves like a narrow path through time. The Y chromosome passes almost unchanged from father to son. That makes it unusually useful for tracking ancestry. Not culture, not language, just descent. When researchers compare Y chromosome markers across ancient samples, they aren't asking who these people thought they were. They're asking where their paternal ancestors came from. In the case of the 7,000-year-old Siberian individuals, the first comparisons produced a result that felt almost reassuring. Their Y chromosome lineages clustered with groups known from Western Eurasia, populations linked to regions stretching from Eastern Europe through parts of the Near East. In other words, the paternal signal pointed west. This matters because it creates a sense of structure, a geographic anchor. It tells us that at least some of the male ancestors of these Siberian individuals belong to lineages that were once widespread far to the west of where the remains were found. That alone already breaks a clean east-west divide, but it does so in a way that still feels explainable. One lineage, one direction, one story. When geneticists visualize this, they don't see names or labels. They see clusters, groups of Y chromosome markers that consistently appear together across individuals. These clusters act like fingerprints. If two individuals share enough of the same markers, they likely share a relatively recent common paternal ancestor. And in this case, the clusters from ancient Siberia overlap strongly with clusters previously identified in ancient Western Eurasian populations. This is where many simplified explanations stop. It becomes tempting to say, a group of men moved east, settled in Siberia, and left their genetic mark. End of story. But that conclusion only holds if you ignore everything else. The Y chromosome tells a focused story, but it's also a limited one. It represents a single thread in a much larger fabric. And even within that thread, the details matter. First, the paternal line doesn't appear as a single, isolated event. The signal isn't sharp and singular. It's distributed. That suggests ongoing connections rather than a one-time migration. If this were a brief incursion, we'd expect a narrow genetic footprint. Instead, we see continuity across samples and time. Second, the geographic interpretation isn't as simple as Europe moved east. The Y lineages involve trace back to populations that existed before modern Europe or modern Asia, as concepts even existed. These were mobile hunter-gatherer groups occupying a vast belt of Eurasia, connected by river systems, open steppe, and seasonal movement. The paternal line doesn't tell us where these men identified themselves. It tells us where their deep ancestry connects. To understand why this matters, Imagine the landscape 7,000 years ago. No borders, no nations, vast territories, with low population density. Movement wasn't exceptional, it was necessary. Groups followed animals. They followed water. They followed opportunity. In such a world, paternal lineages could travel far without implying conquest or mass replacement. A few generations of movement, intermarriage, and resettlement are enough to carry a Y chromosome thousands of kilometers. This is why geneticists treat the paternal signal as an anchor, not a conclusion. It stabilizes the timeline. It gives us a directional hint, but it doesn't explain the whole mechanism. Visually, this is the part of a story that works well on a map. You can draw a broad western region. You can draw arrows extending eastward. You can place a time marker, several millennia before recorded history. And for a moment, the story feels complete. That feeling is important and dangerous, because if we stopped here, we would walk away with a neat narrative that feels satisfying but incomplete. The paternal line gives us clarity, but it also hides complexity. It compresses centuries of interaction into a single directional signal. And there's another limitation. The Y chromosome represents only one person per generation, one male line out of many. Entire family networks disappear from view if they don't pass through that narrow channel. Mothers, daughters, side branches, all invisible in this dataset. So the paternal line answers a specific question very well. Did some of the male ancestry of these Siberian individuals originate from populations further west? Yes, clearly. But it does not answer a more important question. How did these populations actually form? To get closer to that answer, geneticists look for consistency. If paternal ancestry points west, does maternal ancestry do the same? If ancient DNA as a whole reflects the same pattern, the story holds. If it doesn't, something more complicated happened. And this is where the first crack appears. Because when researchers shifted focus away from the Y chromosome and began analyzing mitochondrial DNA, the maternal line, the geographic signals stopped lining up so neatly. The anchor that felt so solid suddenly looked partial. This doesn't invalidate the paternal data. It reframes it. The Y chromosome gave us a clean entry point into the story. It told us that long-distance connections across Eurasia were real and measurable. But it also set a trap. The illusion that ancestry moves in a single direction, carried by a single group. That illusion doesn't survive the next step. If we were to judge these ancient Siberian populations using only paternal data, we'd say the investigation is nearly finished. The movement seems clear. The explanation feels stable. But the moment we bring the maternal line into the picture, the narrative stops cooperating. And that's not a flaw in the data. That's the data doing its job. Because real population history is not built on one line of descent. It's built on mixing, settlement, isolation, and reconnection, processes that affect men and women differently over time. So the paternal line gives us our anchor, but it also tells us where not to stop. If the Y chromosome were the whole story, ancient Siberia would look genetically straightforward. It doesn't. And the reason becomes unavoidable in the next block, when the maternal line enters the investigation, and the map starts pointing in more than one direction at once. Now we look at mitochondrial DNA, and the geography changes. This is where the investigation stops feeling comfortable, because the maternal line does not simply confirm what the paternal line suggested. It complicates it, quietly but decisively. Mitochondrial DNA is passed from mothers to all their children, but only daughters pass it on. That makes it a record of maternal ancestry that behaves very differently from the Y chromosome. It tends to preserve local continuity. It reflects long-term residence, family structure, and who stayed while others moved. When researchers analyzed the mitochondrial DNA of these 7,000-year-old Siberian individuals, they expected alignment. If paternal ancestry pointed west, maternal ancestry should, at least broadly, do the same. It didn't. Instead, the maternal line revealed a different geographic emphasis. In several cases, the mitochondrial signatures aligned more closely with populations that had deep roots in northern Eurasia and parts of Europe, not as migrants passing through, but as groups that had been present and stable over long periods. At first glance, this looks like a contradiction. European-associated paternal ancestry. Maternal ancestry that does not follow the same path. But this isn't a conflict in the data. It's a record of how populations actually formed. What we're seeing is not two incompatible stories. We're seeing two layers of the same story, recorded by different people. The most parsimonious explanation is mixing, but not mixing as a vague concept. Mixing with direction, timing, and social structure. Groups of men moving into regions where women were already present. Over generations, paternal lineages travel long distances, while maternal lineages remain geographically anchored. This pattern is not unique to Siberia. It appears repeatedly in ancient DNA across Eurasia. The mechanism is simple, even if the outcome looks complex. Small groups of men migrate. They integrate into local populations. Their Y chromosomes spread rapidly. Maternal lineages, meanwhile, change more slowly. They preserve the genetic memory of the region. This creates a population that looks contradictory only if you expect ancestry to behave symmetrically. But ancestry isn't symmetrical. The paternal line records mobility. The maternal line records continuity. Once that distinction is clear, the data stops looking strange and starts looking precise. This also explains why labeling these individuals as European or Siberian misses the point. Their genetic identity is layered. It reflects movement layered onto stability, connection layered onto place. To make this intuitive, it helps to think about language. Languages spread unevenly. A new language can arrive with a small group of people and become dominant, even while much of a local culture remains intact. Vocabulary shifts faster than traditions. Grammar survives longer than accents. DNA behaves in a similar way. Paternal lineages can spread quickly and visibly, like a dominant language. Maternal lineages persist quietly, like local pronunciation that doesn't disappear even when the language changes. Neither tells the whole story alone. There's another factor here that often gets overlooked: time depth. These patterns didn't form overnight. They emerged over hundreds and thousands of years. Each generation diluted the original signal slightly, but also preserved it. That's why ancient DNA is so powerful. Modern populations carry too many layers of mixing to isolate these processes clearly. Ancient individuals give us snapshots, moments where the structure of ancestry is still visible. In some of these Siberian samples, the maternal lineages show signs of what geneticists call founder effects. A small number of maternal ancestors contributing disproportionately to later populations. This doesn't imply anything dramatic or violent. It reflects isolation, small group size, and limited gene flow in certain regions. Founder effects come with consequences. Some genetic traits become common, not because they are advantageous, but because they happen to be present in the founding population. In rare cases, this can increase the frequency of certain inherited conditions. This is not a story about pathology. It's a reminder of scale. When populations are small and isolated, chance matters. Genetics preserves that chance long after the context is forgotten. And this is where the maternal line becomes essential. Without it, we might mistake movement for replacement. We might imagine waves of people erasing what came before. The mitochondrial data shows something different: continuity under change. Men arrived, lineages mixed, but local ancestry did not vanish. That realization changes how we interpret the paternal anchor from the previous block. The Y chromosome no longer represents a dominant population replacing another. It represents integration into existing communities. This matters for how we think about ancient Eurasia as a whole. It wasn't divided into sealed regions. It was a connected landscape, with gradients of ancestry rather than borders. And Siberia, far from being isolated, was part of that network. By now, the simple story has fully broken down. We no longer have one direction, one origin, or one explanation. We have a process, movement interacting with place, recorded differently depending on which genetic line we follow. And if this pattern appears in ancient Siberia, a region often imagined as remote and peripheral, it raises a larger question. What happens when we look at populations that were even more isolated? What happens in regions where geography limits movement, where cultural boundaries persist for centuries, and where genetic mixing follows different rules? Because if two lines of ancestry can diverge this clearly in Siberia, the effect elsewhere may be even stronger. That's the direction this investigation has to go next. Once you see how differently paternal and maternal lines behave, it becomes impossible to treat any population as genetically simple. And this is where the story expands beyond Siberia. Because Siberia is not unique. It's just visible. When researchers apply the same methods to other regions, often described as peripheral or isolated, the same pattern appears, but shaped by local geography and history. In parts of East Africa, South Asia, and East Asia, genetic data shows long-term continuity layered with external input, rather than replacement. Take communities in the Horn of Africa. Genetically, many show strong local ancestry stretching back tens of thousands of years. But within that continuity, there are clear signals of contact with populations from the Near East. These signals don't erase local identity. They sit on top of it, like sediment layers recording interaction rather than conquest. In parts of South Asia, the picture is similar. Paternal and maternal lines often tell different stories about timing and direction. One reflects mobility across steppe and plateau. The other preserves deep regional roots. Neither is more true than the other. They answer different questions. East Asia shows yet another variation. Geography channels movement. Mountains, rivers, and coastlines shape contact differently. Genetic signatures reflect that structure, gradual diffusion rather than abrupt shifts. Across all of these cases, the same principle holds. Genetic ancestry is local and connected at the same time. This is why identity cannot be reduced to DNA alone. Genes record biology, not belief. They tell us where ancestry flowed, not how people understood themselves. Culture, language, ritual, and shared memory operate on different timelines. A community can maintain a stable identity while its genetic makeup slowly changes. It can adopt new ancestry without losing continuity. Ancient DNA makes that clear in a way historical texts never could. This brings us back to Siberia and to the European genetic components found there. Those individuals were not anomalies. They were not out of place. They were the expected result of a world where human groups interacted constantly across open landscapes. The surprise isn't that European-associated DNA appears in Siberia. The surprise is that we ever imagined it wouldn't. When we step back, a clearer thesis emerges. Human ancestry is a mosaic, not a tree with clean branches, but a network of overlapping paths. The paternal line offers clarity. The maternal line offers depth. Ancient DNA anchors both in time. Together, they show us something simple and uncomfortable. Origins are not singular. And this resolves the loop we opened at the beginning. There was never one clean origin story waiting to be uncovered. What we call origin is a snapshot chosen from a moving process. Textbooks simplify that process because they have to. DNA doesn't. But DNA also doesn't replace history. Archaeology, linguistics, and cultural memory remain essential. Genes tell us where people came from biologically. They don't tell us who people believed themselves to be. Identity sits at the intersection of these things. DNA plus history plus memory. Remove any one of them, and the picture collapses. The finding of European-associated DNA in 7,000-year-old Siberians doesn't rewrite human history. It corrects its scale. It reminds us that the ancient world was larger, more connected, and more fluid than modern categories suggest. And once you accept that, many other puzzles stop looking strange. The question is no longer why distant populations share ancestry. The question is why we expected them not to. If you want to explore more DNA stories that challenge simple ideas of origin and identity, subscribe.